Thank you. You can all hear me okay? Okay. Well, it's a great pleasure to be here, and many of you know it's somewhat like home for me, so coming back, so it's great. I'm really happy to be here. So I'm going to talk today about a particular study that we've carried out, my group as well as a lot of collaborators over the last 3 years. And again, I think it's kind of an interesting story. I want to tell it that way as a mystery. We started with a fundamental understanding about indium arsenide materials and how to exploit these materials, their advantages for electronic devices. And we moved into a new area, which is surface-based sensing, which we had not had a lot of experience in, and then solution-based sensing for biomedical applications. And we found a lot of interesting results that showed us that things aren't always the way they appear to be. The mystery is the story I'd like to tell you, how we figured out what was happening and how we can take advantage of what was happening. So I think this is sort of how we untangled this, and as kind of an outsider in a field can see things in a different way and take advantage of it, something that a lot of you in your careers will come up against at various times. I have a huge number of collaborators that were all crucial to this work in different ways: chemists, chemical engineers, MD, PhD student I had, people in the Institute of Genomic Science and Policy at Duke. So I thank them or acknowledge them. So I'm going to start by talking about the particular project that spawned this work. A lot of this is generalizable, but there's some that's specific to this project. It was a DARPA-funded project, and it was interesting because we, the people making sensors, there were 3 groups making different types of sensors. We're trying to make sensors for point-of-care applications, which is an application of interest for many different medical conditions and sites for use. But in this particular case, it was interesting because the idea was to be able to basically identify biomarkers that told you that you were going to get sick before you had any symptoms. So, for example, they tested this against flu, and they were able to identify a set of about 15 mRNA that were expressed at higher concentrations in approximately the picomolar concentration range, many days before symptoms arose. The reason DARPA was interested in this is that they wanted people in the field, soldiers, to be able to be tested and then sequestered if they were going to get sick. So it was interesting in that way. So we were trying to build a sensor that could be used in the field, and again could test at these concentrations a set or an array of biomarker elements for mRNA that could easily be turned out for different conditions, so we could test for different specific mRNA. Like all point-of-care applications here, there are many technical challenges. You'd like it to be very, very sensitive, have a large dynamic range, again, because we didn't know at what concentrations we'd have to test, multiplex measurements, very rapid time to response. Ideally, they wanted 5 minutes. Of course, instantaneously would be perfect. Minimal pre-analyte processing, so taking blood, a small amount of blood, minimal processing. So this is about the biggest challenge you can have in a biomedical sensor application. So I show here again, what are the key challenges? These are all the challenges that related primarily to the sensor, again, meeting the performance requirements, sample complexity, sample volume, and then these were the related project challenges. That is, were the biomarker really good choices, and could this information be used? This is always a question in biomedical sensor applications. Is the information gained from the sensor of clinical utility? Can decisions be made? So moving to the general class of our device, there were 3 teams making sensors. One group was using a fluorescence-based sensor, one was a SIRS-based sensor, and we were making an electronic sensor. The basic diagram of how the sensor performs is shown here. This is an affinity-based binding sensor that's been exploited many times, a very old idea. So you take the idea of having some sort of transducer here, attaching a molecule that has an affinity to what you want to capture and sense. So for example, this would be an antibody for a protein sensor. It could be a DNA or a PNA for mRNA or DNA, et cetera. So the challenge is to make this sensitive, that is the transduction mechanism, to attach the molecules appropriately, and to be able to pull out what we want from this complex mixture shown here. So we chose indium arsenide, and it will become clear to you as I go through the slides why we chose indium arsenide. And that is a critical decision. It's a compound semiconductor, but it has specific properties that were of interest to us. We realized high sensitivity in a planar device geometry. That is, the sensitivity we realized was as good as nanowire sensors, but we did not understand why. The challenges in this are molecular surface functionalization. We'll show the importance of the oxide at the surface of this material in mediating chemical reactions and how we can build on this idea. As I said, electronic sensors that are a conductance-based sensors have been explored for decades. I showed this slide because I want to point out this is work by Art Janata at Georgia Tech, who is really a pioneer in this field and articulated all of the challenges and different implementations. The basic idea here again with a FET-type sensor of any sort is the conductance changes in the channel, the receptor layer and the binding acts as a molecular gate. So that when binding occurs at this molecular gate, which is selective, it's the affinity, it's a probe that has a specific target to bind to, this potential or charge will change on the surface, thereby changing the conductance of the channel. It's a very, very simple concept. There are two primary challenges. One of these is that in real samples at physiological conditions, you're going to have salt in the sample, and so there can be charge screening in these solutions. Okay, so that causes the charge here to be screened by ions here, which will dampen the sensitivity or the change in the conductance. The other is that when you have a normal FET channel, say you had silicon or gallium arsenide here, the electrons are distributed throughout this three-dimensional volume. You're, for example, depleting this volume by the gate potential here. But again, there's a relative insensitivity to what happens at the surface. Okay, so ideally you'd like to be able to place the molecules as close as possible to the channel and have the channel be as narrow as possible, again, so that you modulate it effectively. It's a similar problem to trying to scale field effect transistors down to very small sizes. So it's a scaling problem. Nanowires, sensors, electronic sensors here, pioneered by Charles Lieber. And this shows some results early on, and I think this was, I can't see the year here, 2004, results shown here. Solved the second problem very well, and it's simple to understand why nanowires do that. Here's a nanowire with current conducting through the center of the nanowire with molecules all around the surface, so a very large surface area to volume. The modulation of this charge is much more effective. And so just looking at adsorption of a molecule, in this case DNA, on the surface of a nanowire, this gives an idea of the sensitivity. DNA concentrations down to low femtomolar concentrations yielded conductance change here, shown here, and what corresponds to the lowest DNA concentration of 10 femtomolar here, this was about a 3.8% change in the conductance, to give you an idea of the numbers. So it was a breakthrough, and it was also important in that you could do real-time monitoring. So this is in solution-based sensing. You have a specific conductance, you expose the nanowire to the target in solution, and you can see the conductance change in response to exposure or to binding. So we started out fairly naively. Again, I'll get to why the interest in indium arsenide, but I first want to show you the results that we achieved. Proceeding naively, we attach molecules, we do some tests, we see how well it works. That's the way things are done quite a bit. So similarly to the nanowire results that I just showed you, this shows DNA concentration in solution. So this is the molarity of the solution. We use many different types of DNA. This was single-strand DNA, 25 bases. And this shows now in this case, we're measuring electron density. So instead of conductance, we could do a Hall measurement in the indium arsenide, and we could measure the electron density and the electron mobility separately of measurements of the impact of the binding on the surface. So this shows the initial value, and this shows the change after absorbing from this concentration solution over 24 hours. And what we found is immediately is that this is very sensitive. I mean, just like the nanowire result, 10 femtomolars, this was almost 6% change. You can see it seems to increase if you look at the percentages here, but don't look too close. It's part of the mystery. That it almost looks monotonic, but we'll come back to that.
So,
yes?
How do you measure the electron?
Hall measurement. Hall measurement. So we take the sample before, we do a Hall measurement, that's the baseline. We measured after functionalization of the surface, after. That's very simple. Could not be more simple. We also had experience with indium arsenide sensors in the gas phase to build upon, and we knew that they were sensitive for these conditions as well. This is work in which we functionalized the surface of indium arsenide with porphyrin molecules. This was iron-centered porphyrins. They attach by the carboxylic acid groups here. And this shows gas phase sensing of nitric oxide, which has a chemical affinity to the iron center of the porphyrin. And so we got responses in the low parts per billion range, which is very, very good, very highly sensitive. Again, I'm just showing you that we got good initial results. Mike Angelo was my MD PhD student, and he was looking at antibody sensing. This is in physiologic solution, so this was in about, I think it was 200 millimolar PBS solution, so a lot of salt in the solution. And this shows the response functionalizing with antibodies and then exposing, for example, the anti-TNF molecule covered sensor to TNF alpha. And we have a response here in comparison to a control where it was exposed to HSA. So you can see that the pairs matched up. The conductance change in this case, or the density change, was much higher. Again, the only point to this is to say, gee, things seem to work, and they seem to be fairly simple, but that's not the case. What are these structures like? We're doing Hall measurements. Again, they're simple structures. We had about 3 generations of these simple devices. The first, look at this. This is just 4 contacts on the corner of indium arsenide on a slide with a little quartz tube beaker. That's where we put the solution so it would sit. Then we would rinse it several times, dry it off, and measure it dry. That was first generation. For the gas sensors, this just shows we measured conductance here, the resistance of this pattern at the end of a PCB board mounted sample. And then for all the sensor results I'm going to show you for the DNA sensing, we used a Hall bar structure shown here. And so we could actually have 2 areas for sample exposure. The size of this was in the 10s to 100 micron range size. So we could expose here, dry the sample off, and then measure a Hall measurement after. Simple structures. So I promised to tell you why indium arsenide, and this tells you a little bit more about that. Here's a diagram of indium arsenide. Very simple. It's grown on gallium arsenide. I don't show the substrate. That's not really important to the story here. It has a native oxide that I'll talk more about that just grows in air naturally. And it has, because the Fermi energy is pinned above the conduction band in indium arsenide. This is a surface potential, so the conduction band, this is the Fermi energy. There is a native 2-dimensional electron gas shown here that occurs right at this interface, that exists right at this interface. The nature of this 2-dimensional electron gas and what it arises from is part of the mystery of this story. But this is what we're measuring, is the electron density and mobility of this 2-dimensional electron gas. A little bit more results shown here. One important thing is that just like in an accumulation layer, inversion layer in a MOSFET, there's a relationship between the electron density in the 2-dimensional electron gas and the mobility. In the range we were in, which was density concentrations in the 10 to the 11 to 10 to the 12, to even some in the 10 to the 13 per centimeter squared range. We're in a range where the mobility, the scattering results primarily from surface potential lateral non-uniformities and from charge at the interface. So this is the relationship here due to that scattering. This just shows more data again, looking at the density change. Here we were using thiol attachment. Here we were using carboxylic acid attachment. That is, we were taking DNA to make the probes. We were modifying the end of the DNA so that we could enhance the attachment of the probes. Why that's important will become clear to you. I'm foreshadowing in this mystery. So I mentioned that the concept for how these sensors work could not be more simple. But in fact, even while there are lots of very good sensor results published, when you actually try to compare the results to what is expected from a simple theory, they don't work very well. That matching doesn't occur very well. And there's some very nice work by Ron Nyman at the Weizmann Institute on how these sensors really work and what some of the challenges are. The idea of using a two-dimensional electron gas that's right at the surface to enhance sensing sensitivity is not a new one. Although in this case, they looked at using a gas-algas two-dimensional electron gas in which they grew it and butted it up against the surface. In this case, it didn't work so well. You may not see this so well, but basically the change in conductivity, even when the electrons are placed right against the surface, was virtually zero at room temperature and was only measurable at very low temperature, 5 K. But there's a little bit there at 100 K. So the question is, why is the sensitivity relatively poor in structures like this? The fundamental coupling mechanisms are unclear, but they're considered to be of 3 different physical mechanisms. One is when the electron binds to the surface, there's a charge transfer that changes the potential. One is that a dipole exists on the surface. This dipole can be the dipole of the molecule itself, and if they're arranged all in the same conformation and they have a strong dipole moment, then that will change the electron affinity or the work function. And one is that surface states are modified by the attachment. It turns out to be this latter one is what's crucial. And it's also the more difficult one to think about. How do we attach molecules to the surface? This is very important to any of these processes. Again, what we're trying to do first is attach the receptor. So we want to attach a short single-strand DNA that's going to bind to its complementary DNA that's the target. So we have to think about a number of things. What is required for attachment? How does the functionalization process impact the surface? What is the degree of chemical passivation? And this has been looked at over and over. So if you attach molecules to all available sites on the surface and you've removed the oxide before, then a measure of the effectiveness is that the surface becomes chemically stable. All the sites are bound by molecules, and so oxidation does not reoccur. So chemical passivation is considered important. What is the impact on surface electronic properties? And related to this chemical passivation is stability. For the 3,5 materials, there's been much less work than on metal surfaces, for example, or on silicon. Much of the semiconductor work builds on the very successful functionalization or attachment of molecules to gold through the thiol functional group. So many people started with the 3,5s looking at thiols, considering, hey, that should work also. Well, not really. Also phosphonic acids and carboxylic acids. We looked at all of those, well, directly or indirectly. So after functionalization, the question is, how do you need to characterize and determine the following parameters? What is the density and ordering of the molecular overlayer? What is the conformation of the molecule? If it's DNA, for example, if it's attached by one functional group, is it vertical? And are all of the molecules, or does it have some inclination, average angle? What is the electrostatic environment created by the surface? If you consider hybridization, then hybridization is a very complex process where the targets come in near the probes. How far apart the probes are is crucial. What the electrostatic environment is very crucial. And then what is the nature of the solution interactions? That is the hydrophobic forces at the surface. All of these are important. getting down to more specific details in the 3,5s here, this is the primary approach that's been used for the 3,5s. Remove the native oxide. That's always number one. Functionalize. You may or may not use a linker. That is, you may functionalize. We did with, again, with the porphyrin or heme molecules. We did not have to attach a linker because the carboxylic acid groups bound well to the 3,5 surface. Create a self-assembled monolayer if we're lucky, which means that it's ordered, it's high density, it's chemically passivated, and again, we can characterize it. The oxide turns out to be crucially important, as we'll show, and generally for the 3,5s, the phosphate binds more strongly than the carboxylic acid, which binds as a carboxylate, is much greater than the thiolate. And so this shows the mechanism of the binding of the phosphonic acid. And it shows here that basically this binding happens through a condensation reaction where hydroxyls that terminate the surface here are consumed. In this case, there was an oxide, are consumed, and we have a covalent bond between either arsenic or gallium, oxygen, and the phosphorus. Characterization, we did a lot of characterization. There are a number of approaches we looked at, and again, one of the reasons I show you these next three slides is to show the importance of controlling the density on the surface. This slide shows our use of spectroscopic ellipsometry to determine through a model the relative thickness of the molecular overlayer. And also you want to understand the kinetics of the attachment. So this shows the thickness of the heme molecule attached to indium arsenide, functionalized for over 3 hours, it saturates, and so you have possibly a monolayer under those conditions. The other thing that's important is to look at the concentration of the solution. And for example, with porphyrin molecules, you can get aggregates that form, so you need to optimize there as well. So it's a fairly complex process, and what's really important is to be able to control the concentration of molecules so that you minimize nonspecific binding, which is the undesired molecular interactions with the surface that lead to a response. You want selective sensing. It decreases sensitivity, yields false positives, and impedes the ability to quantify the sensor response. And you can see how this happens. This shows a test using ellipsometry of functionalizing a surface with anti-HSA, that should be, human serum albumin. And we had 15 to 20% coverage in this case and 100% coverage in this case. When this changes upon exposure to BSA, which is not its complement, it shouldn't bind, this shows that binding does occur when we have only a 15 to 20% coverage. So the BSA is interacting non-specifically with the surface. But here when we have good coverage, we have 100%. Probably wasn't exactly 100%, but much better. And we could not have a measurable response here. So that's very important for the sensor application. So now let's move to the DNA, to the specific studies that I want to talk about here. And I'm going to show you the results from two different tests. One are just looking, again, at the free adsorption of single-strand DNA. How does that change the surface? And one is to do a hybridization test. Functionalize, then expose the surfaces to the complementary solution, as well as a control, which would be junk DNA in solution to determine what the nonspecific binding response should be. We did a lot of different characterization shown here. I'll go into it. We looked at two different types of DNA. This is DNA. The groups that we're interested in binding would be the phosphate carbon backbone. The phosphate backbone, we know that that will interact, as well as the carbonyl and the amine groups in the bases, as well as the conjugated base rings as well, can all interact. So we looked at one, we have modified carboxylic acid, so we had a carboxylic acid group down on the end here. That shows a thiol modification, but that's what we had, as well as unmodified DNA. And I'm showing you back the same data again. I'm taking you back now to the beginning. So just a replot of we adsorbed this concentration of DNA on the surface. We get this percentage change in the electron density of the surface. So what's important is this looks very well behaved. It almost looks too well behaved. I mean, I don't have error bars on here, but they would be in the, plus or minus 5% for these levels, okay? But I didn't put in one data point. I left one out. This was the mystery for a long time. Now I put the data point back in. It's the blue one right here. So not only was this electron density, but it was also surface potential and other methods of measuring. We saw this same strange behavior around the micromolar concentration range. So we carried out another experiment, and there are many replicates of all of these, in which we looked at this very narrow concentration range around here, and these are the results we got. So I'm going to talk about these results. Why is this happening? Well, to understand why this is happening, we have to understand the nature of the oxide at the surface. And to do that, first we need to talk about using XPS very briefly, because all of our chemical characterization is using X-ray photoelectron spectroscopy, in which we can look at the binding of all the elements. The elements we want to look at are the oxide chemistry, so we have indium oxide and arsenic oxides. Okay. We want to also look at binding, so we'd like to be able to look at the elements in DNA. We can look at the nitrogen 1s core level shown here. It's different for different bases. We can look at phosphorus core level. We can look at carbon. I'm not going to go into a detail about all of this, but I want to talk about how we used it. And we can determine a lot about the binding by putting sets of data together in a complementary fashion to understand what was happening. We can also look at the valence band spectrum and band bending. We can determine the Fermi energy, and there are ways of supporting this through other measurements as well. What's important is we can't really separate, there's oxygen in DNA, we can't separate the oxygen from the oxide oxygen, except at the very extremes of binding energies. So we do that to some extent. So go back to the data, and we carried out a measurement using XPS of a means of assessing the relative coverage of the DNA. So this again covers this more narrow range from 0.25 micromolar to 4 micromolar. And I've expanded this. And a way of assessing the relative coverage is to take the total nitrogen 1s core level in XPS, the area, integrated area, and to divide it by either a reference, indium or arsenide, in the substrate. And you can quantify this by carrying out a number of calculations, known attenuation depth of the electrons, photoelectrons, et cetera, for the different elements. And these coverages are in the 10 to the 12 per centimeter squared range. So they're reasonable coverages. Very high coverages would be in the 10 to the 13. They're reasonable. And what this shows you is that this looks pretty well behaved for some of the points, but there are these definite outlying points where the coverage is much higher. How do we understand that? Well, we need to go back and look at the oxide. And this shows the phase diagram for the oxide that's going to form on indium arsenide. The important elements here, and now when I talk about oxide elements, I will talk about these elements as if this is a compound and has, for example, In2O3, which is the indium oxide, and different oxygen states for arsenic oxide here, as well as this ternary oxide. But what's important to understand is that this is really bonding configurations. So it's the density of relative bonding configurations that exist. Okay, so it's really hard to separate them out, but we do that in order to understand what we're seeing. The oxide is typically in this thickness range, 25 to 30 angstroms or so. And again, the point is multiple oxides exist with different chemical features. And what we knew, because we carried out a study of thiol attachment on this surface, is that first of all, if you etch the surface of indium arsenide or gallium arsenide in different ways, for example, this just was looking at etching, looking at the native oxide here and etching with different solutions, and you vary the takeoff angle in XPS. So 90-degree takeoff angle means you have normal incidence, and your normal incidence of the X-rays, and a smaller takeoff angle is getting more shallow probing of the surface. So this indicates what's happening more towards the surface. This is a little bit deeper, and none of them are very deep. Think 100 angstroms or so, okay? So this says, well, the arsenic to indium ratio, as well as the oxide components, when we look at the indium and the arsenic core levels, are changing significantly, where here we have a higher arsenic oxide oxidation state for the native oxide, and then it's etched for the hydrofluoric solution etch much differently for the sodium hydroxide than from the sodium hydroxide. So the preparation of the oxide depends very much on the chemical treatment. That shouldn't come as a surprise. And we had worked on thiol attachment for some time. There was a mystery in terms of thiol attachment for indium arsenide and gallium arsenide. People engaged in this field could not understand why it looked like there was thiol attachment to gallium sometime with gallium arsenide and arsenic at other processing conditions and times, and the same for indium arsenide. So what we did is look at changing cysteamine concentration, and cysteamine has a thiol end group, so we were looking at that attachment, varying the concentration of cysteamine, and looking at either samples where the native oxide exists, or we had an etched surface. And what we found is that the concentration of relative bonds, indium disulfur and arsenic disulfur, changed dramatically with concentration and also depended upon the oxide. The model that we developed and talked about, I won't go into too much detail, basically said that the rates of reactions in this interaction between the thiol and different arsenic oxide oxidation states, the rates of attachment and dissolution of those phases changed with concentration. This concentration was the limiting reactant in a number of different chemical reactions that lead to this changing attachment. So that first gave us the idea is that we needed to think about what happened with the surface as a chemical reaction between the oxide as a reactant that has different phases and the molecule as a reactant and the solution. The solution's important here because functionalization for us, we carried this out 24 hours, 20 microliter sample, humid environment, light, so it was in water. Hybridization was in water for 30 minutes. The reactions can be quite varied. And the best model we had for these reactions actually were gleaned from environmental chemistry research. So looking at the weathering of minerals in water, at looking at arsenic remediation in natural waters and how arsenic interacts with surfaces provides a lot of information about how to think about this problem. So there's oxidation can occur, reduction of the oxides can occur, water can dissociate at the surface as well as dissociate the different components or bonds in the oxide. pH changes occur with our very small volume. We could have maybe large pH changes right at the surface, which will change the reactions. And we have, again, complex formation, adsorption, and then dissolution. So ligand-enhanced dissolution of oxides is well understood, well, relatively well understood, in this literature. But it's an area of very active research. So we went back and looked at our samples now and said what happens around 1 micromolar concentration. And we looked at the indium oxide and arsenic oxide in our samples. And it's pretty easy to see what's happening. As we get close to this concentration range, the indium oxide is decreasing dramatically. The arsenic oxide is decreasing as well, although what we'll see is the arsenic oxide is changing. We can see reoxidation quite a bit, dissolution. And so arsenic oxide really controls what's happening at the surface, but the indium oxide is crucial. So more about this. So the oxide is dissolving, and the concentration of the DNA changes the rate of this dissolution reaction. So we're having attachment, hydrolysis of some of these bonds, and then some detaching, taking away, dissolving the oxides as it happens. We look at the pore bay diagram. This is for gallium arsenide to understand what solutions we'll have in species. We have to understand that as well. So the arsenic oxides can be in these different degrees of protonation at the surface, which will change the reactions. For indium oxide, like gallium oxide, We are well below the point of zero charge, which is around 9, pH of 9. We're at near neutral pH. We have hydroxyl termination of the indium. This will likely be positively charged. The indium oxide does not dissolve readily unless it's unless it is terminated by a hydroxyl group, which then dissolves. So the higher concentration of hydroxyl groups cause a higher rate of dissolution. All of these are important. We did a lot of studies just looking at what happens in water with bare or molecule covered surfaces. Arsenic oxide dropped within 15 minutes significantly in water. The different oxidation states change with time. DNA coverage can be reduced by 50% in 15 minutes if you put it into water. pH slight shifts, but we can't really measure what's happening near the surface. But the change in two-dimensional electron gas density was relatively small given these large changes. And one thing we did is understand, so what is the role of carboxylic acid? All of these functional groups are important in determining rates of reactions. This is a little bit complex, but you can understand this if I step you through it. This shows indium oxide. On the y-axis, it's exposure to DNA with carboxylic acid groups and unmodified here. If you ignore the blue points for a minute and just look at the open circles, we took the same concentrations and we just plotted the indium oxide concentration for each of these. Okay? We can see for the open circles, we don't have a very nice trend. But if we offset the concentration of DNA for the carboxylic acid groups by 0.25 micromolar, it lines up much better. Same for the arsenic oxide. If we offset it by 0.5 micromolar, we get better agreement. And so this tells us that the carboxylic acid enhances arsenic oxide dissolution. It's equivalent for an unmodified DNA with a 0.5 micromolar higher concentration. And for indium oxide, there is a protective action by the binding with the carboxylic acid that reduces the dissolution equivalent to 0.25 micromolar, roughly. As we're going through this work, many people are asking us, why do nanowire devices work so well? Why should we be looking at this when everything is dissolving? It's not stable. This is well known. And so a month or two ago, a paper by the Lieber group just came out in Nano Letters indicating, well, there is an issue with stability, even of silicon nanowires. So this is a problem. This is a problem in general, for solution-based sensing. And so how you deal with it is important. In this case, the idea was to actually protect the surface through a shell around the nanowire, but of course that's going to decrease your sensitivity, but it's important. Going back to our data again, back to this data, coverage versus DNA density. What are these? Why do we have these high points here, high concentrations? Well, if we plot the coverage for both modified in red and unmodified DNA, it really fits very well with the indium oxide percent. So you can see that we're attaching in proportion to the indium oxide. And then if you look at the Fermi energy at the interface of the indium arsenide, the indium oxide is controlling the Fermi energy. We can measure by extrapolating the front leading edge of the valence band spectrum in XPS, the Fermi energy. And we can ensure that this measurement is good because it's tricky, this assessment, by looking at core level shifts. So we have to do that. And we can see that basically the band gap of indium arsenide is down at 0.35 eV. This is EF, this is the Fermi energy minus the valence band. So these are all well above the conduction band, as we expected, well above the conduction band, and again, dependent upon the indium oxide, which there's been speculation in the literature. I've read a lot of old literature going back with this to understand oxide formation, etching. There are a lot of papers that say oxides control the Fermi energy, but not a real good mapping between them. And there's been a lot of recent work on understanding what is the cause, what defect or what property of indium arsenide leads to the two-dimensional electron gas, because that's what we're controlling. This is work by Chris van der Waal here in 2010 saying what are the causes of electron accumulation layers on indium arsenide surfaces. And again, these have not been identified. It's known that there must be some surface donor that's an intrinsic defect donor. And the two candidates from his models are indium add atoms on the surface that are at 0.78 EV and hydrogen on the surface. Going to go through this kind of quickly, but there are numbers of papers that look at similar things. Indium arsenide is interesting as well because the Fermi energy is above the conduction band and other properties. It is considered the dominant 3,5 material that will work well for MOS applications in the future. And there are a lot of papers coming out now showing that the surface can be inverted, the Fermi energy and controlled. This means the defect density is low. So there's a lot of work to understand the nature of these defects. John Robertson carries out a lot of this work. This shows, again, indium dangling bonds here are dominant defects in the conduction band, and arsenic-arsenic bonds, arsenic binding to each other. So this is free arsenic that results from oxidation or reduction of the oxide. There's also been a large body of literature in the physics community.
In your samples, what is the thickness of the indium arsenide layer?
Varied, but typically maybe 100 angstroms, well, 100 to 500 angstroms. Or thicker, even 1,000. They vary.
Is it pseudomorphic or relaxed?
It's relaxed. They're all relaxed.
So you can have you do have a huge density of defects.
Yes, we would have, yes.
So do you know what those defects, what kind of role they might play?
We basically consider them in terms of the electronic properties because we did do a lot of the chemistry experiments also on bulk indium arsenide substrates.
So it's similar?
Yes. So as far as that chemistry mapping, yeah. There's also been a large body of works in the surface physics community using XPS to understand the nature of the two-dimensional electron gas and how it's modified by adsorbates. And so this is related as well. Shown here that just with less than 0.1 monolayer adsorption of many different elements on the surface can change the Fermi energy quite a bit. And so it's an area of interest in many different fields. And again, we wanted to look at how the Fermi energy changes. This is just from a couple of samples here. That is, for experiments, do we have binning? This shows basically a histogram of where the Fermi energy sits for different measurements. Do we have binning? Do we have defect levels that show up more and more? And so we're in the process of understanding this better now. It relates somewhat to what's in the literature, but we're not quite there yet. So let's go back. I need to compress a little bit here. This is the electron concentration range with DNA adsorption for the same experiments. This shows functionalization over initial, so it's about a 25% change here, 15% with the difference shown here. 25 compared to the initial condition here. And we're going to focus a bit on, so what can we learn about the binding? Why does this density change? We know the oxide changes. How does the DNA bind? And we're going to focus a bit on these two samples, one with the lowest concentration change and one with the highest concentration range. They're only separated by 0.25 micromolar DNA concentration. And what we found is that we could readily, this is the electron density, we could readily relate the electron density to the phosphate backbone binding. So this core level energy indicates a different binding environment for the phosphorus, and it directly relates to the electron density. I don't show you the data here, but the substrate oxygen, the oxide, also shifts with this, so it indicates that we have binding between phosphorus and oxygen in the surface. Now understand that this is on average because we're going to have mixtures of bonding types. So this means that on average for this sample we have much more phosphorus binding. When the phosphorus core level moves to lower energy, it becomes relatively more negative. The oxygen becomes relatively more positive because now we have just a redistribution of charge in that bond. And again, with metal oxides, there's a large body of work on understanding these core level shifts in binding. And the mobility depends on conformation. This is a way of determining something about average conformation. If I take the phosphorus to nitrogen, or one of the core level peak fits, and I take just one measurement and take that ratio, it should look like the stoichiometry of the DNA. That is, What is the number of nitrogen atoms in the DNA with the different bases versus the number of phosphorus? But if you take the 60-degree takeoff angle to 90-degree takeoff angle, and it changes, it means that as this number gets larger, the phosphorus is enhanced in the surface of the DNA. That means on average, it's not bound down at the interface. It's a little simple-minded, but it works. Again, it becomes a piece of the puzzle. And what it shows is that as the phosphorus becomes less bound to the surface, the mobility increases. And this is good because we just said as the phosphorus binds more, the density increases. So this is all consistent. Two different ways of assessing the same thing, that phosphorus binds to the surface under certain conditions and increases the electron density. And this shows we can measure the change in mobility over density, and that depends on the coverage of the DNA. Okay, so it's a different measure as well. Here, we can see this relationship with the coverage of the DNA. To understand a little bit more about what was happening with the binding, we used IR spectroscopy. There's been a large body of work done there on just different attachment to metal oxides, but DNA analysis as well. I show you a little bit of FTIR here, and we're focusing again on these two samples. The lower density concentration at 0.5 micromolar and the higher at 0.75. It's very different. Here, the red is 0.75 where the phosphorus is flat on the surface binding. And this is the asymmetric vibration for the phosphorus oxygen in the backbone. This again indicates for the 0.5 micromolar solution it's more vertical. This is more flat, which is consistent. This shows the methylene groups in the backbone. There's a dramatic density here. So we actually have an ordered crystalline-like state depending on these vibrations for the 0.5 micromolar. So it's not lying down flat, but it's very ordered. And we can look at the frequency for the hydroxyl groups and how they shift. And we can understand that for this 0.5 micromolar sample, we have much more hydrogen bonding. So this ordered structure has a strong hydrogen bonding network on the surface. This shows the indium oxide or arsenic oxide enrichment on the surface. This is indium oxide enrichment. This is arsenic oxide enrichment. The difference between these two is the arsenic oxide of the surface. When we have the hydrogen bonding, we have a high arsenic oxide at the surface. Probably the indium has dissolved into solution, okay? And here we have a low arsenic oxide, and that completely changes the binding. Again, consistent with what we showed here. So it's these dissolution reactions, oxidation reactions that depend on concentration that cycle and change the conformation. So nitrogen also, we discovered, binds to the arsenic as well. And we did this by looking at core level shifts, and we understand this to be an electrostatic bonding because of the way the energy change for the amine group and the state of protonation of the arsenic oxide. So that's probably the second dominant bonding mechanism. I want to quickly go through hybridization. I just have a few here. So we expose samples to both target in blue and control in red. This shows the coverage for over a range of 8 femtomolar to 0.8 micromolar. It's a huge range. You can see here we have higher coverage when we have the target to the probe. So this is good. It means we have hybridization, and that enhances the coverage. And it also changes the oxide as we thought. When we have high coverage, we have low arsenic oxide. When we have low coverage, we have high arsenic oxide. And it changes the Fermi energy in a controlled way. Fermi energy is shown here for control and target over a range in the femtomolar range, actually, and it relates to the indium enrichment or depletion in the oxide as well. So coverage and conformation go together again, and we can infer something about the conformation here, again through looking at the nitrogen to phosphorus ratio. And we can see that when we have just nonspecific binding, that is, we don't have good hybridization, we have more amine arsenic oxide interaction, basically. Now, can we measure this electronically? We had to understand the chemistry. And we're still working on this to some extent, but we can see if we take, these are small changes. This range I'm looking at here is in 10 femtomolar to 1 nanomolar range. It's a very small DNA concentration range. And it says the difference in target to control in the change in electron density scales with the arsenic oxide, which makes sense to us, but we can read it out electronically. And this is the mobility change as well. Not quite as good a fit, but it fits with what we saw before. So it depends on the arsenic oxide. In this case, in fact, it was related more to the specific oxidation state of the arsenic oxide. And this is my last data slide, so I'm almost at the end here. You can carry out Hall measurements under light and dark conditions. So you have 4 parameters. Under light or dark conditions, you are differentially populating states in the sample. And this actually has a robust response that will probably lead to good sensing. So target minus control, if we take the ratio of light and to dark density and mobility, this scales directly with the free arsenic, the arsenic-arsenic bonding that's supposed to be the most important defect in MOS structures in this material. And we can see that we can use this for sensing under those conditions. So what is the concept that we came up with at this point? Ultimately, we'd want to design oxides for a specific sensor. But the idea shows here. Think of the surface as a chemical template that we can electronically read out or control. So we have molecule semiconductor chemical reactions. These drive, and there's feedback here, molecular conformation through binding, defect stoichiometry changes, and the 2-dimensional electron gas mobility, and mobility, density and mobility. And so our findings are shown here that indium arsenide possesses this sensitivity, again, because of the nature of the 2-dimensional electron gas. That's really its electron affinity. While unstable from a standard device use, that we may get higher functionality if a sensor is designed to be a chemical switch for a conformation change or at a specific density, and we should be able to make highly sensitive devices. So in conclusion, we hopefully can exploit these characteristics. We can measure 2 to 4 parameters for robust detection at low density. And we can ultimately think about electronic control as well, because the electron availability in the oxide and the surface can be important for chemistry. And that's it.